Imagine walking into a massive modern poultry house, right just before dawn.
Oh, yeah. It's a very specific vibe.
It really is. The air is deliberately warm. It's hovering right around 90 degrees for the young chicks.
Right, because they need that heat.
Exactly. And the lighting is low, it's soft, entirely designed to keep the birds calm.
Yeah.
You look out over this sea of wood shavings, and there are, 30,000 young chickens just resting quietly on the floor.
It sounds incredibly peaceful.
Right. To anyone walking in off the street, everything looks perfectly peaceful. It looks like the absolute pinnacle of modern, healthy agriculture.
It does look peaceful, on the surface.
Yeah.
But the reality is that agricultural environments are these incredibly complex ecosystems.
Right.
And that peace, well, it is almost always an illusion.
It is, because beneath the feet of those 30,000 birds, just hiding microscopically in the warm, moist wood shavings on the floor, is this invisible enemy.
Yeah, an absolute killer.
It's a tiny single-celled killer, actually, and it's just waiting for the perfect moment and the perfect environmental conditions to strike.
And when it finally does strike, the biological and the economic damage is just catastrophic.
It's massive.
We aren't talking about a minor nuisance for a local farmer.
Right.
This specific organism creates a multi-billion-dollar disaster year after year.
Yeah, it fundamentally alters how global agriculture operates.
It really does.
So, welcome to our deep dive. Today, we're taking on a massive topic in basic poultry science. We are pulling from a really heavy stack of veterinary textbooks, scientific reviews, and academic papers.
We're going to explore one of the most expensive and honestly, most devastating diseases in the entire global poultry industry.
We're breaking down the biology of how this disease works, right? The ecology of how it spreads through a commercial farm and the incredibly complex chemical warfare that farmers use to fight it.
It's a lot of ground to cover.
It is. Now, I want to make a quick note for our listeners. We know many of you use this show to practice your technical English.
Right, building up that professional vocabulary.
Exactly. You might even be practicing a language technique called shadowing, where you listen to a phrase and repeat it out loud to master the pronunciation.
Which is a fantastic way to learn, by the way.
It is. So today, we're deliberately slowing our pace just a bit. We're keeping our sentences a bit shorter.
Yeah, we want to make sure the science is totally accessible.
Right. We're using clear language to explain the science. And when we hit a crucial technical term, we'll define it simply. We will give you a real-world example of how it's used on a farm, and we will leave a brief, natural pause in the conversation.
So you can use that pause to repeat the word out loud.
Exactly. We want you to fully grasp the concepts and the vocabulary, get comfortable, listen closely, and practice speaking along with us.
Because the science we're looking at today is, well, it's incredibly intricate. Taking our time to clearly define the mechanisms is going to help everyone.
Whether you're an English learner or a first-year veterinary student. Okay, so let's unpack this. Our sources note that this single disease costs the global poultry industry over $3 billion every single year.
$3 billion. It's a staggering number.
It really is. But before we can understand how farmers fight this enemy, we have to name it. We need to understand exactly what it does to the animal.
And that brings us right to our first core concept. The medical condition we are discussing is called coccidiosis.
Right.
Coccidiosis is a common and devastating intestinal disease in livestock, especially in chickens. Coccidiosis.
Coccidiosis. Now, here is how you might hear a veterinarian use it in the field. If a flock has bloody diarrhea and poor weight gain, the farmer must test for coccidiosis.
I will give you a second to practice saying that at home. Coccidiosis.
Awesome. Now, when we talk about coccidiosis, we are talking about the clinical disease, right?
Yeah, it's the actual physical condition of the sick bird.
Right. But the disease itself is caused by a very specific biological agent.
Exactly. The disease is the symptom, but the pathogen is the cause.
And that pathogen is our second key term today.
Mm-hmm.
The organism causing all this destruction belongs to a genus called Imeria. Imeria is the genus of the parasite that causes the disease. It is a single-celled protozoan. Imeria.
Imeria. In a diagnostic report, it might sound like this. Imeria tenella is a specific parasite that attacks the ceca of the chicken.
Go ahead and try saying that one out loud. Imeria.
Great.
Now, I want to stop and look at that word protozoan for a second. Because, when people hear single-celled, they immediately think of bacteria.
Oh, sure. That's the default assumption.
Right. But Imeria isn't a bacteria. And
No.
it certainly isn't a virus.
Not at all. Bacteria are prokaryotes, which are much simpler cells. Eimeria is a eukaryote.
Meaning it's much more complex.
Highly complex. It's a single-celled parasite with a nucleus and sophisticated cellular machinery. It actually belongs to a group called the Apicomplexa.
Okay.
It's actually related to the parasite that causes human malaria, which gives you an idea of how formidable it is.
Wow. Okay, so it's a serious pathogen. And the sources emphasize that Eimeria is incredibly host-specific.
Very much so.
What does that specificity actually look like in nature?
Well, it means the parasite has evolved this biological lock-and-key mechanism. It only fits one type of animal.
Just one.
Just one. The Eimeria species that infect chickens will absolutely not infect a cow. They won't infect a pig.
Okay.
They are genetically hardwired to recognize and invade only chicken tissue.
So if a farm has mixed poultry, like, say, turkeys and chickens living near each other, a turkey can't catch coccidiosis from a sick chicken.
Correct. The turkeys are perfectly safe from chicken Eimeria.
Oh, that's fascinating.
But, turkeys have their own evolutionary enemies. They get sick from highly specific turkey parasites, like a species called Eimeria miliogrammitis.
Right.
Meanwhile, chickens are battling species like Eimeria tenella, Eimeria maxima, or Eimeria acervulina.
Okay, let me try an analogy here to map this out, because there are a lot of species names.
Go for it.
I like to picture the chicken's digestive tract as a complex car engine.
Okay, I like that.
An engine has distinct parts with distinct jobs. You've got the fuel line, the air filter, the exhaust manifold. Do these different Eimeria species attack different parts of the engine?
That's actually a brilliant way to conceptualize it.
Yeah.
Yes, they do. The host specificity goes even deeper than just the species of the bird.
Really?
Yeah, it targets specific geographical real estate inside the bird's gut.
How specific are we talking?
Extremely specific. Let's look at Eimeria tenella, for example.
Okay.
If a chicken ingests Eimeria tenella, that parasite travels through the stomach and the upper intestines without doing anything at all.
It just floats on by.
Just floats on by. It waits until it reaches a very specific section called the ceca. The ceca are two blind pouches located near the end of the intestinal tract where fermentation happens.
Okay, so it waits for the ceca.
Right. Eimeria tenella recognizes the cells there, it invades them, and it causes severe, life-threatening bleeding.
So if the gut is an engine, Eimeria tenella is like rust violently eating through the exhaust line, right? Causing a massive catastrophic leak.
Exactly. But then you have Eimeria acervulina.
Okay, another species.
Right. This species ignores the ceca completely. It targets the upper part of the small intestine, known as the duodenum.
And what does the duodenum do?
The duodenum is where the bird absorbs the vast majority of its nutrients from its feed. Eimeria acervulina doesn't cause massive hemorrhaging.
No blood.
No, instead, it systematically blunts and destroys the tiny finger-like projections called villi. And those villi are what absorb the food.
Wow, so that's like a completely clogged air filter or a broken fuel injector.
Yeah, exactly.
The engine might still be running, but it's completely starved of what it needs. The chicken is eating, but the nutrients are just passing straight through.
And that biological reality explains a vital economic point in our sources. You might assume that the $3 billion price tag of coccidiosis comes from dead chickens.
I mean, yeah, dead birds cost money.
Right, but mortality is actually just a fraction of the cost.
Wait, really? A disease this destructive doesn't primarily cost money through dead birds.
Dead birds are an obvious loss. But according to the data, roughly 80% of that $3 billion cost is entirely hidden. It's categorized as subclinical loss.
Subclinical, meaning the bird doesn't look sick to the naked eye.
Right, the bird survives. But because its intestinal lining has been ravaged, because the engine is clogged, its feed conversion ratio plummets.
The feed conversion ratio, or FCR.
Yeah.
That's a massive metric in farming.
Oh, it's everything.
It's basically how many pounds of feed it takes to grow 1 pound of chicken, right?
Exactly. Feed is by far the highest cost in poultry farming. If a chicken with a subclinical Eimeria infection is eating expensive, high-quality feed, but its destroyed gut can't absorb the protein, that feed is just wasted into the litter.
The farmer pays for the fuel, but the car doesn't go anywhere.
Yes. And over 30,000 birds, that lost efficiency just bankrupts farms.
Man. So now we know what Eimeria is, and we know how it financially bleeds a farm dry by destroying the digestive engine. But how does this microscopic parasite actually move from one bird's gut to infect an entire flock of 30,000?
Well, that takes us out of the bird and onto the floor of the poultry house. We need to look at the environmental stage of the parasite.
Okay.
And this brings us to our third essential term, oocyst.
Oocyst.
Right. The oocyst is the egg-like stage of the parasite. It is equipped with a tough outer wall that protects it in the environment. Oocyst.
Let's give you an example of the term in context.
Yeah.
The sick chicken sheds millions of oocysts into the litter on the floor.
Take a quick second to say that one. Oocyst.
Excellent. Now, I should clarify, you said egg-like.
Right, we call it egg-like to help visualize it, but biologically it is microscopic cyst.
Got it.
And that tough outer wall is an absolute marvel of natural engineering. It is incredibly resilient.
How tough are we talking? Like, can a farmer just spray bleach on the floor and kill them?
No. That's what makes Eimeria such a nightmare. The oocyst wall is highly resistant to mechanical damage, and it is largely impervious to standard chemical disinfectants like bleach or iodine.
You're kidding. Bleach doesn't work.
Doesn't work. The oocysts can sit in the wood shavings, in the dirt, and in the feces for months, completely shielded from the outside world.
Wow. But, just sitting in the dirt doesn't cause a disease. The oocyst has to find a way inside a healthy host.
Right.
Which leads us to our fourth technical concept. This describes the pathway of infection. It's called the fecal-oral route.
A very descriptive term.
It is. The fecal-oral route is how the disease transmits. It occurs when an animal eats something contaminated with infected feces. Fecal-oral route.
You might hear it discussed in a biosecurity context like this. The disease spreads through the fecal-oral route when birds peck at dirty litter.
Go ahead and try saying it. Fecal-oral route.
Good.
Now, when you think about chicken behavior, the fecal-oral route is almost unavoidable.
Oh, completely unavoidable.
I mean, chickens are natural foragers. They spend their entire days scratching at the ground, pecking at the litter, looking for spilled feed or insects.
It's their fundamental biological instinct. In a modern poultry house, the birds are living directly on top of their own litter.
Right.
When they peck, they're inevitably ingesting microscopic particles of the environment, and that includes the oocysts left behind by other birds.
But the sources reveal a really strange twist here, and I want to make sure I get this right.
Okay.
If a healthy chicken pecks the ground, right, and eats a fresh oocyst that was just passed by a sick bird a minute ago, nothing happens.
Nothing happens.
The healthy bird doesn't get sick.
No, it is entirely harmless at that exact moment.
Mm.
A freshly shed oocyst is biologically dormant. It is completely uninfective.
Really?
Yeah, it requires a specific incubation period outside the bird's body to become dangerous.
And that external incubation period is our fifth crucial term. It's called sporulation.
Sporulation.
Sporulation is the developmental process outside the host where the dormant oocyst transforms and becomes infective. Sporulation.
Let's use this in a practical farm management sentence. Wet litter speeds up sporulation, making the oocysts dangerous to the flock.
Say that out loud with us. Sporulation.
Great job.
So what exactly is happening during sporulation? Why does the outside environment matter so much?
Well, it's basically a biological metamorphosis.
Yeah.
Inside that tough wall, the single cell divides into multiple spores, which are called sporocysts.
Right.
And those sporocysts, in turn, hold the actual infective agents, the sporozoites. But the oocyst will absolutely refuse to do this unless it senses the perfect environmental triggers.
What triggers does it need?
It requires three absolute conditions: oxygen, a specific warm temperature, and moisture.
Oxygen, heat, and moisture. So if the floor of the poultry house is perfectly dry, the oocyst just sits there, completely dormant.
Exactly. It enters a state of suspended animation.
Yep.
But, poultry houses are full of living, breathing animals.
Right, 30,000 of them.
The birds generate heat, they exhale moisture, they excrete water in their waste. If the farmer's ventilation system isn't perfectly managed, the wood shavings on the floor become damp and warm.
It's honestly like setting the timer on a bomb.
I like that analogy a lot. Break that down for me.
Well, think about it. The sick bird drops the bomb, which is the unsporulated oocyst, into the litter, but the bomb isn't armed yet. It doesn't have a fuse.
Right.
It just sits in the dirt. But the moment the weather conditions in the barn change, the moment the litter gets warm and wet, the bomb's internal clock starts winding up.
It goes through sporulation.
Exactly. And once that process is finished, the bomb is fully armed and lethal. It is just waiting for a healthy bird to swallow it.
That is precisely how it functions. And under optimal warm and wet conditions, that arming process, sporulation, takes anywhere from 24 to 72 hours.
Just 1 to 3 days.
Fast, right?
Yeah. Okay, so the bomb is armed. The healthy chicken pecks the wet litter and swallows the sporulated oocyst. What actually happens inside the bird to set the bomb off?
This is where the mechanics of the chicken's own body basically work against it.
Huh.
The oocyst travels down the esophagus and enters the bird's digestive system.
Okay.
Now, chickens don't have teeth, they don't chew their food.
Right.
Instead, they have a highly specialized, incredibly muscular stomach organ called a gizzard.
Right. The scientific term is the ventriculus. The gizzard is incredibly powerful. Chickens actually swallow small stones and grit, which sit in the gizzard and act like millstones to physically grind up hard grains and seeds.
Exactly. The gizzard applies tremendous mechanical crushing force, and that tough, armored wall of the oocyst, which survived bleach and the outside environment, is violently crushed and cracked open by the chicken's own gizzard.
The armed bomb has been breached?
Yes. And breaking that outer wall releases the next stage of the parasite, which we call the sporozoos. These are microscopic, highly mobile, missile-like cells.
Missile-like.
Yeah, they travel out of the gizzard and swim down into the intestinal tract.
And because of that host specificity we talked about earlier, they swim until they find their specific target real estate, like the ceca or the duodenum.
Correct. Once they arrive, they physically ram their way into the healthy epithelial cells lining the bird's gut. They breach the cell membrane, crawl inside, and completely hijack the cell.
They just hide inside the host's own cells? Do they just sit there stealing nutrients?
Oh, it's much more aggressive than that. Once safely inside the host cell, the sporozoid begins a phase of explosive asexual reproduction.
Okay, reproducing inside the cell.
In parasitology, this is called schizogony or morogony. That single parasite begins copying its nucleus over and over and over again incredibly fast. One parasite turns into a massive cluster of hundreds of new parasites inside that one single intestinal cell.
Wow, the cell must just swell up like a balloon.
It swells until the cell membrane completely fails. The host cell literally explodes, dying instantly.
Jeez.
And those hundreds of newly cloned parasites burst out into the intestinal tract. Each one of them immediately swims to a new healthy cell, burrows inside, and starts the exact same asexual replication process all over again.
This is exponential destruction. Like one parasite destroys a cell and creates 100. Those 100 destroy 100 cells and create 10,000. Those 10,000 destroy 10,000 cells and create 1,000,000.
And that exponential destruction of the cellular lining is exactly what causes the severe internal bleeding, the blunted villi, and the massive drop in nutrient absorption. The bird's engine is being systematically blown apart from the inside.
That is horrifying. How does the cycle eventually end? Do they just keep replicating until the bird dies?
Well, if the dose was high enough, the bird dies from the tissue damage. But the parasite's ultimate goal isn't to kill the host.
Right, it wants to survive.
Exactly. It wants to reproduce and escape. So after a few rounds of this explosive asexual reproduction, the biology of the parasite shifts. It senses the environment and enters a sexual reproduction phase.
Sexual reproduction. So they differentiate into male and female.
Effectively, yes. This phase is called gametogamy. The parasites develop into what are called microgametes, which act like sperm, and macrogametes, which act like eggs.
Okay.
The male microgametes swim through the gut to find and fertilize the female macrogametes.
And that fertilized union creates, let me guess, a brand new oocyst.
Precisely. A brand new, thick-walled oocyst is formed. It is shed out of the bird's body in the feces, landing in the litter, completely unsporulated, just waiting for warmth and moisture to start the entire nightmare over again.
The speed of this cycle is what really blows my mind in these sources. How long does it take from the moment the chicken swallows the oocyst to the moment it poops out 1,000 new ones?
Depending on the specific Eimeria species, the entire internal life cycle takes only 4 to 7 days.
4 days. That is staggeringly fast. If one oocyst turns into hundreds of thousands in less than a week, you can really see how an entire flock of 30,000 birds could be completely overrun almost overnight.
It's a numbers game,
Yeah.
and the parasite is exceptionally good at math.
But, because all of this catastrophic damage is happening deep inside the chicken's gut, it raises a huge practical question for me.
What's that?
If a farmer can't see the torn-up intestines, how do they actually measure the severity of the disease?
Right. You can't manage what you can't measure. Veterinarians have developed specific protocols for this, which leads us to our 6th key term, lesion score.
Lesion score.
The lesion score is a standardized numerical grade. It's given after examining a dead bird's intestines, measuring the physical damage caused by the parasite. Lesion score.
Here is how a farm manager would use the term. The veterinarian assigned a high lesion score because the bird's gut was severely damaged.
Take a moment to practice that one, lesion score.
Perfect. So this standardized system, where did it come from?
The standard system used worldwide was actually developed by Johnson and Reed back in the 1970s. It takes all the guesswork out of the diagnosis.
How does the scoring actually work? Is it just like looking for blood?
It's highly structured. A veterinarian will select a sample of birds from the flock and perform a necropsy, which is an animal autopsy.
Right.
They open the intestinal tract and assign a score from 0 to 4.
I assume 0 being a perfectly healthy, pristine pink intestine?
Correct. No visible lesions at all. A score of 1 means there are a few scattered lesions, tiny spots of damage, but the gut wall looks mostly normal.
Okay, so a mild case.
Right. But by the time you get to a score of 3, the gut wall is severely thickened, there is obvious bleeding, and the contents of the intestine might be mixed with clotted blood.
And a score of 4. The maximum.
A score of 4 is catastrophic. The intestine is swollen, entirely disrupted, filled with massive amounts of blood or dead tissue, and the bird is either dead or actively dying. This is called a gross lesion score, meaning the damage is visible to the naked eye.
But the sources also mention microscopic scoring. Why would they need a microscope if the damage is that obvious?
Because multiple species of Eimeria can infect a bird at the exact same time. The gross lesions tell you how bad the damage is, but a microscopic scraping of the tissue allows the vet to identify the specific size and shape of the oocysts hiding in the cells.
Oh, I see.
That tells them exactly which combination of Eimeria species they are fighting.
Okay, so necropsies tell you what is happening inside the dead birds. But can farmers measure the danger level out on the floor of the barn, like before the birds get sick?
Yes. They take environmental samples to calculate the OPG.
OPG stands for oocysts per gram.
Exactly. A technician will walk through the poultry house and collect small samples of the litter and feces from different locations.
Okay.
They take that dirt to a lab, put it into a special liquid that makes the oocysts float to the top, and literally count them under a microscope to determine how many oocysts are in 1 single gram of litter.
Our sources give a very specific mathematical threshold for this. They note that if the OPG level exceeds 5 times 10 to the 4th power, clinical coccidiosis almost always occurs.
5 times 10 to the 4th is 50,000.
Yeah.
If you have 50,000 microscopic parasites in 1 gram of dirt, the environmental load is so high that the bird's immune systems are simply overwhelmed.
I actually want to push back on that idea for a second. If a farmer gets a lab report back, and the OPG is lower, say, 10,000 or 20,000 oocysts per gram, is that still considered a failure? Does any presence of the parasite mean a disaster is looming?
That is one of the most counterintuitive aspects of poultry science. The answer is actually no. A moderate or low presence of the parasite is not a disaster.
Really?
In fact, it is functionally necessary, because coccidiosis is a self-limiting disease.
Explain self-limiting in this context, because that sounds contradictory.
Yeah, it means the host's own biology can put the brakes on the parasite's reproduction. If a young chick pecks the floor and eats a very small number of oocysts, say a few hundred, the parasites invade the gut and start replicating.
Okay.
The chick might feel slightly lethargic, but it doesn't suffer severe damage.
Because it's a tiny invasion force.
Right, and more importantly, that tiny invasion force triggers the chick's immune system. White blood cells recognize the Eimeria. They learn its signature. The bird develops natural adaptive immunity.
So a mild exposure acts exactly like a natural vaccine.
Precisely. Once the bird has built that immunity, it can swallow thousands of oocysts later in life, and its immune system will instantly destroy them before they can replicate. The disease limits itself.
So, paradoxically, if a farmer builds up a perfectly sterile hospital-grade barn and the young chicks never encounter a single oocyst, that could actually be incredibly dangerous.
It's a ticking time bomb of a different sort. If you raise birds in a hyper-clean environment, their immune systems remain completely naive. They have no defenses.
Oh.
If those birds are suddenly exposed to Eimeria when they're older, maybe a worker accidentally tracks some contaminated mud into the barn on their boots, the parasite will sweep through that flock like wildfire, causing massive mortality.
Because there's no natural immunity to slow it down. The farmer's really walking a razor's edge here. You want enough parasites in the litter to train the immune system, but not so many that it triggers a grade 4 lesion score.
It is a profound ecological balancing act. And because relying entirely on natural immunity is incredibly risky and unpredictable in a barn of 30,000 birds, the global poultry industry has historically relied on chemical intervention.
Which brings us to the defense arsenal. For decades, farmers have mixed specific medications into the chicken feed to control the parasite population. And the most common weapon they use introduces our 7th core vocabulary word:
Ionophore.
Ionophore.
An ionophore is a specific class of antibiotic that disrupts the chemical balance of a parasite cell membrane, causing it to run out of energy and die. Ionophore.
In a feed mill context, you would hear it used like this. The farmer mixed an ionophore into the feed to protect the young chicks.
Let's have you try saying that one. Ionophore.
Excellent. Now, our sources dive deep into the biochemistry of ionophores, and it is absolutely fascinating. These aren't artificial chemicals cooked up in a lab, right? They are naturally occurring compounds.
Yes, ionophores are polyether antibiotics. They are primarily produced by soil bacteria, mostly from the Streptomyces genus. These bacteria evolved ionophores millions of years ago as chemical weapons to fight off competitors in the dirt.
So human scientists essentially borrowed a weapon from bacterial trench warfare.
We did, yeah.
How do these weapons actually kill the Impair parasite? The sources say they disrupt the ion balance, but let's break down the mechanics of what that means.
Okay, let's zoom in to the microscopic level.
Okay.
Picture the Impair sporozoid, that missile-like cell trying to invade the chicken's gut. The outer skin of that cell is a lipid bilayer membrane. It acts like a highly secure border wall.
Right.
Inside the cell, the parasite maintains a very careful specific ratio of salts, specifically sodium and potassium ions.
Okay, I'm picturing a microscopic submarine. The hull is the membrane, keeping the ocean out, and inside the sub, the pressure and chemistry are perfectly regulated.
Perfect analogy. Now, an ionophore molecule is highly lipophilic, meaning it loves fat. It dissolves perfectly into that lipid hull of the submarine.
Okay.
It inserts itself into the cell membrane and acts like an unauthorized open tunnel,
Like a hole in the hull.
a very specific kind of hole. It forcefully binds to sodium ions from the outside environment and shuttles them across the membrane, dumping them inside the parasite. Suddenly, massive amounts of sodium are flooding into the cell.
The submarine is taking on water. Well, salt in this case.
Right. And the parasite's biology instantly panics. It knows that high internal sodium is lethal. It disrupts all cellular function. So the parasite activates its emergency pumps, specifically the sodium-potassium ETPase pumps, to try and push the invading sodium back out.
But running those pumps requires energy.
Massive amounts of energy. The parasite burns through its entire supply of ATP, its cellular energy currency, desperately trying to bail the sodium out of the hull.
Wow.
But the ionophore tunnels are constantly letting more sodium in.
It's a losing battle. The pumps just can't keep up.
Exactly. The parasite exhausts its entire energy reserve. Furthermore, as sodium builds up inside, osmotic pressure changes. Water rushes into the cell to dilute the salt, the cell swells up, and eventually it dies from osmotic shock before it ever gets the chance to invade the chicken's intestinal tissue.
That is an incredibly elegant, brutal weapon, but biology never sits still. Which brings us to the inevitable consequence of chemical warfare and our eighth vocabulary term, drug resistance.
Drug resistance.
Drug resistance happens when pathogens genetically mutate and evolve, rendering the medication unable to kill them. Drug resistance.
This is the dark cloud hanging over the industry. You might hear someone say, because no new drugs have been invented, drug resistance is a massive threat to the farm.
Say that phrase for us. Drug resistance.
Oh,
So it's the classic evolutionary arms race. You apply a chemical pressure, and the survivors pass on genes that can withstand that pressure. But our sources draw a really stark contrast between how resistance develops against synthetic chemicals versus natural ionophores.
yeah. The sources categorize the weapons into two different philosophies. Synthetic chemicals, which are drugs completely synthesized in a laboratory act, like a sniper rifle.
Explain the sniper analogy. How does a chemical act like a sniper?
A synthetic drug targets one very specific microscopic vulnerability in the parasite. For example, there's a class of synthetic drugs called quinolones.
Okay.
They don't mess with the cell membrane. They go straight for the mitochondria, the powerhouse of the cell. They bind to one extremely specific protein in the electron transport chain, shutting off the power supply.
Highly precise. One shot, one kill.
Extremely lethal. But because the mechanism is so precise, relying on a single structural protein, it is very easy for the parasite to defeat it.
Really? How?
Well, if the parasite undergoes a random genetic mutation that slightly changes the physical shape of that one mitochondrial protein, the synthetic drug can no longer bind to it.
The sniper's bullet just bounces off, the lock changed, so the key no longer works.
Exactly. And because Impair replicates by the millions in a matter of days, that one lucky mutant quickly becomes the dominant strain. Resistance to synthetic sniper chemicals builds incredibly fast, sometimes within a single season.
But ionophores are different. The sources compare them to artillery.
Right. Ionophores don't target one specific protein lock.
Mm.
They fundamentally alter the thermodynamic and osmotic pressure of the entire cell membrane. They blow gaping holes in the basic physical chemistry of the organism.
It's much harder to evolve a defense against artillery. You can't just change the shape of one protein to stop an explosive change in osmotic pressure.
Precisely. Evolving resistance to ionophores requires complex multi-gene mutations to completely redesign the cell membrane. It's biologically expensive for the parasite.
Right.
Therefore, ionophores have remained effective for decades, whereas synthetics burn out quickly.
But the sources also highlight a brilliant secondary feature of ionophores. They describe them as leaky.
Yes, ionophores do not provide sterile immunity. They are not 100% lethal to every single parasite. They kill the vast majority of the Eimeria, but a tiny percentage leak through the drug's defense and manage to replicate in the bird's gut.
At first glance, a leaky drug sounds like a defective drug. You want to kill all the enemies.
Not in this ecosystem. Remember our discussion about the self-limiting nature of the disease.
Ah, right. Because the drug leaks, a tiny, non-lethal amount of the parasite survives. The chicken's immune system detects that small surviving population and builds natural antibodies against it.
Exactly. The ionophore suppresses the massive fatal infection, but allows a microinfection to occur. It works in tandem with the host's own immune system, resulting in lifelong natural immunity.
That is absolutely critical for birds like breeder hens or egg layers, right, who live for a long time and need durable immune defenses.
It's a masterpiece of biological management.
It really is. But I see a huge red flag in the sources regarding ionophores. They have a very narrow safety margin.
That is the trade-off with artillery. A narrow safety margin means the dosage that kills the parasite is uncomfortably close to the dosage that poisons the host.
So if a feed mill technician makes a math error and adds just slightly too much ionophore into the mixing vat, it won't just kill the parasite, it will kill the chickens.
And it's not just a danger to the chickens. The sources point out that ionophores can be lethally toxic to non-target species. The most famous example is monensin.
Monensin.
Yes, monensin is a widely used ionophore that is safe and effective for chickens at the prescribed dose. But it is profoundly toxic to equines.
Horses.
Yes. If a horse accidentally gains access to a bag of chicken feed medicated with monensin, the results are tragic. The drug attacks the horse's cardiac muscle, causing severe, often fatal, heart failure.
Oh, wow.
And it's species-specific, too. An ionophore called selenomycin is perfectly safe for broiler chickens, but if you accidentally feed it to turkeys, it will wipe out the entire flock.
So farmers are wielding incredibly powerful, potentially toxic tools, and the parasite is constantly trying to evolve past them.
Yes.
And because the pharmaceutical industry hasn't released a fundamentally new class of anti-coccidial drugs in decades, the industry has to carefully manage the aging weapons they have left.
That brings us to the strategic logistics of preserving drug efficacy. To slow down resistance, farms implement complex rotational schedules.
Which introduces our 9th vocabulary term, shuttle program.
Shuttle program.
A shuttle program is a management strategy where a farmer changes the specific type of anti-coccidial medication given to a single flock during its lifetime. Shuttle program.
Here's how a farm manager explains it. To stop the parasites from adapting, the farm uses a careful shuttle program.
Take a second to practice this one. Shuttle program.
Nice. To understand a shuttle program, you really have to understand the lifespan of a modern commercial chicken.
Okay.
A broiler chicken, which is grown for meat, typically lives for about 6 to 8 weeks before going to market.
And over those 6 weeks, their nutritional needs change rapidly. So the farmer feeds them different diets. Week 1, they get a high-protein starter feed. Later, they get a grower feed. And finally, a finisher feed.
Right. A shuttle program means changing the medication every time you change the feed phase. In a classic shuttle program, a farmer might put a synthetic chemical, the sniper, into the starter feed for the first 2 weeks.
This cleans out the barn, hitting the early Eimeria population hard and fast with high precision.
But we know that if you leave the sniper in the feed for too long, the parasite mutates.
Right.
So at week 3, when they switch to the grower feed, they completely remove the synthetic chemical and replace it with an ionophore, the artillery.
I see. By switching weapons mid-cycle, the parasite population gets conceptually confused. Any parasites that were beginning to mutate a defense against the sniper are suddenly blasted by the artillery. They never get the time needed to establish a resistant genetic lineage.
Exactly. Now, it is important to contrast a shuttle program with a rotation program.
What's the difference?
A shuttle program changes the drug during the life of one flock. A rotation program changes the drug between entirely different flocks.
Okay, so in a rotation program, flock A moves into the barn and eats ionophores for their entire 6-week life. Then flock A goes to market, the barn is cleaned, flock B moves in, and flock B is fed synthetic chemicals for their entire life.
Correct. Both are valid strategies to deliberately alter the chemical pressure and break the parasite's evolutionary momentum.
Makes sense.
But even with these programs, drug resistance is creeping up. Furthermore, global consumers are increasingly demanding meat raised without chemical antibiotics.
The no antibiotics ever movement. This market pressure is forcing farmers to look for natural alternative weapons, which brings us to our 10th term, phytogenics.
Phytogenics.
Phytogenics are plant-based feed additives containing natural extracts and essential oils used to support gut health and inhibit pathogens. Phytogenics.
You will see this heavily advertised in modern farming. Many antibiotic-free farms use phytogenics to help keep the birds healthy.
Let's hear you say it. Phytogenics.
Excellent.
The sources mention several specific plants used in these phytogenic blends, like oregano, cilantro, and artemisia, which is sweet wormwood.
These plants produce powerful secondary metabolites. Oregano, for example, is rich in compounds like carvacrol and thymol.
What do those do?
These are phenols that can physically disrupt the cell membranes of bacteria and, to some extent, parasites. Artemisia produces artemisinin, which generates reactive oxygen species that damage the parasite internally.
That sounds incredible. I mean, it's natural, it's safe, and it attacks the parasite. Why aren't we just replacing all chemicals with oregano oil?
Because biology is rarely that simple. And here we must introduce a very serious warning found in our academic review sources.
Oh, a warning.
The reviewers explicitly caution against scientific overreach.
Overreach, meaning promising more than the data can actually deliver.
Exactly. The academic reviewer analyzed dozens of published papers on phytogenics and alternative treatments. They found a disturbing trend of scientists and companies using absolute, definitive language that completely ignores the messy reality of biology.
Give me an example of overreach. What does that sound like?
Well, the reviewer highlighted a paper that claimed a specific phytogenic product has been proven to greatly reduce mortality. That phrasing is a huge red flag.
Why?
It implies a guaranteed outcome. It treats the plant extract like a magic bullet.
But why is that scientifically inaccurate? If oregano oil kills parasites in a petri dish, why doesn't it guarantee survival in the barn?
Because of the baseline microbiota. A petri dish is a tightly controlled environment. A chicken's gut is a chaotic jungle of billions of interacting bacteria.
Right.
If a flock of chickens already possesses a highly balanced, robust microbiome, adding a phytogenic might not show any measurable improvement.
It's already healthy.
Exactly. Conversely, if a flock is under severe environmental stress and facing an overwhelming OPG load of 50,000 oocysts, a dose of oregano oil is simply not going to save them the way a potent ionophore would.
The plant extracts modulate the environment. They don't sterilize it. They are supplements, not silver bullets.
Exactly. We must avoid absolute language. And the academic reviewer applied this exact same critique to the literature surrounding coccidiosis vaccines.
Vaccines are interesting. Instead of drugs or plants, you spray a controlled dose of live, unattenuated oocysts onto the chicks to manually trigger their natural immunity.
It's a very effective strategy.
Yeah.
But the reviewer found papers claiming that a vaccine program provides complete coverage or ensures patient safety.
That's overreach again.
Yes, because avian ecosystems are stochastic.
Stochastic, meaning inherently random and unpredictable.
Right. When you spray a liquid vaccine over 30,000 scrambling chicks, the distribution is never mathematically perfect.
Some get more, some get less.
Exactly. There will be spatial clusters where some chicks receive a heavy dose and others miss the spray entirely. Furthermore, immune systems fluctuate based on temperature, stress, and genetics. Claiming complete coverage in a barn of 30,000 living, breathing animals is scientific arrogance.
Realistic, probabilistic expectations are mandatory.
Very much so.
That is a phenomenal reality check. So up to this point, our entire deep dive has been focused on Impair. We've been talking about an intestinal parasite that uses the fecal-oral route, hiding in the litter and blowing up the digestive engine.
But our sources don't stop there. They pivot to highlight an entirely different class of parasites that attack poultry in a fundamentally different way.
And because the attack vector is completely different, the farmer's defense strategy has to change completely. This brings us to our 11th and final vocabulary word, vector.
Vector.
A vector is a living organism, usually an insect, that carries and transmits an infectious pathogen from one host to another. Vector.
In a biosecurity plan, it sounds like this. To stop avian malaria, the farmer must control the mosquito, which is the vector.
Try saying it out loud. Vector.
Perfect.
So we are shifting our focus to a disease complex known as avian hemosporidiosis. Hemo meaning blood.
Yes, these are blood parasites. The sources list 3 primary genera: Leucocetozoon, Plasmodium, which is the pathogen responsible for avian malaria, and Hemoproteus.
Unlike Imaria, these parasites don't care about the chicken's intestines. They target the red blood cells and the internal organs, like the liver and spleen.
And most critically, they completely bypass the fecal-oral route. You cannot catch avian malaria by swallowing dirty litter. The pathogen must be injected directly into the bloodstream.
Which is why they rely entirely on vectors.
The sources identify the specific culprits. For Leucocetozoon, the vectors are black flies from the Similidae family.
Okay.
For Hemoproteus, it's biting midges from the Culicoides family. And for Plasmodium, it's mosquitoes.
The mechanics here are so sinister. The insect lands on a wild infected bird, maybe a pigeon or a wild duck. It bites the bird and drinks the blood, ingesting the parasite. The parasite actually undergoes a part of its reproductive cycle inside the insect's gut.
Yes, the insect isn't just a taxi, it's a required biological host. Then the insect flies into the poultry house, lands on a healthy chicken, bites it, and directly injects the infective stage of the parasite into the chicken's capillaries.
This fundamentally changes the rules of the game for the farmer.
It changes everything about biosecurity. If you are fighting Imaria, your biosecurity is focused on the ground.
Right.
You manage the litter moisture, you change your booze to avoid tracking in feces, and you use anticoccidial feed.
But none of that matters against a mosquito. Managing the floor doesn't stop an insect flying through the window.
Let's bring back your earlier analogy. How does this change the warfare?
Well, defending against Imaria is a ground war. The enemy is in the dirt, acting like landmines. You defend the floor. Defending against Plasmodium is an air raid. The enemy is coming from the sky.
A very apt description. To fight the air raid, the farmer has to look outside the barn. They have to eliminate standing water where mosquitoes breed.
Right.
They have to manage the brush and vegetation around the farm where midges hide. In endemic areas, they literally have to install specialized insect-proof netting over the massive ventilation fans.
And the sources point out a fascinating genetic divide in how birds handle these blood parasites. They compare native backyard chicken breeds to modern commercial high-yield breeds.
It's a stark lesson in genetic trade-offs. Native village chickens breeds that have lived outdoors in tropical or subtropical environments for centuries have evolved alongside these insect vectors.
Their immune systems have adapted to the constant presence of avian malaria. They might get bitten, and they might carry the parasite in their blood, but they have a natural tolerance. They rarely die from it.
But modern commercial broilers are genetic marvels, engineered for one specific purpose, and that is rapid, efficient muscle growth. They have been selectively bred over decades inside highly controlled, vector-free indoor environments.
They are pampered super athletes, but the sources note that if you take those commercial high-yield breeds and try to raise them in an open, free-range environment where these vectors are active,
the results are devastating. The sources state that exposure to these blood parasites in commercial breeds can result in up to 90% mortality.
90%, 9 out of 10 birds dead just from mosquito bites.
It proves that when we intensively select an animal for extreme production traits, like a massive breast muscle and fast growth, we often inadvertently sacrifice their robust ancestral immune capabilities. A commercial broiler simply does not have the genetic hardware to survive an air raid.
Wow. We have covered an immense amount of scientific ground today. We started in the microscopic valleys of the intestinal tract and ended up fighting mosquitoes in the sky.
We really did.
We explored the evolutionary arms race of chemical snipers and ionophore artillery and the delicate balance of the poultry house ecosystem.
To our listeners practicing their English today, you've tackled some deeply complex biological and agricultural concepts. Let's do a rapid-flowing recap of the 11 core terms you shadowed, locking in those definitions one last time.
Number 1, coccidiosis, the devastating clinical intestinal disease that costs the industry billions.
Number 2, Imaria, the specific genus of the highly targeted single-celled protozoan parasite.
Number 3, oocyst, the microscopic armored capsule that survives in the dirt.
Number 4, fecal-oral route, the behavioral pathway of infection caused by pecking at contaminated litter.
Number 5, sporulation, the biological arming sequence requiring moisture, warmth, and oxygen.
Number 6, lesion score, the standardized numerical grading of physical intestinal damage from 0 to 4.
Number 7, ionophore, the natural polyaether antibiotic that acts like artillery, altering cell membrane pressure.
Number 8, drug resistance, the evolutionary mutation that allows pathogens to survive our chemical weapons.
Number 9, shuttle program, the strategic rotation of drugs during the life of a single flock to confuse the parasite.
Number 10, phytogenics, plant-based extracts like oregano used to modulate the gut, recognizing the danger of scientific overreach.
And number 11, vector, the living, flying organisms like mosquitoes that transmit blood parasites, requiring entirely different biosecurity.
Incredible work today.
Before we sign off, I want to leave you with one final provocative thought drawn from the deeper implications of our sources.
We've established that Eimeria is successful because of its math. It relies on the sheer density of the host.
The sources refer to it as the crowding effect. Modern commercial farming puts 30,000 birds into a single closed envelope. That architectural density is what allows a single parasite to multiply into an environmental disaster in a matter of weeks.
For 70 years, we've used chemical additives to artificially suppress that math. But resistance is inevitable. The drugs are failing, and consumers are rightfully demanding we stop relying on them.
So here's the question to ponder. If biological resistance eventually forces us to permanently abandon our chemical arsenal, can the current model of the poultry house survive? Or will the fundamental architectural design, the density, and the entire economic structure of how we raise food have to be entirely reimagined just to outrun the biology of one ancient microscopic parasite?
It is a profound question that the next generation of agricultural scientists will definitely have to answer.
Something to think about the next time you drive past a quiet, peaceful farm. Thank you for diving deep with us today. Keep asking questions, keep practicing, and we will see you next time.